welcome to today's webinar. My name is Eugenia and I'll be your host. It's our pleasure to have Solvias with us today, who will be presenting this webinar titled Milligrams to Kilograms, Discovering Optimal Solid Forms of Drug Substances. Today's guest speaker is Susan DePaul, Ph.D. Susan is the team leader of the Solid State Development Group at Solvias AG in Switzerland. Her team specializes in contract research in the field of solid-state development of pharmaceuticals. This includes discovering crystalline forms, polymorphs of drug substances, crystallizing multi-component systems, salts, co-crystals, characterizing the physicochemical properties of these solid forms to guide preformulation, selecting the best solid form for development, and developing a crystallization process from the milligram to kilogram scale. A physical chemist by training, Susan received her Ph.D. from the University of California, Berkeley, in the field of solid-state NMR. She has over 20 years of experience in the crystallization of pharmaceuticals, holds approximately 10 patents, and has co-authored several chapters and books, most recently Chapter 14 in Polymorphism in the Pharmaceutical Industry, Solid Form and Drug Development. Now I'll provide some housekeeping information before we start. First of all, I would personally like to welcome you to our webinar platform on24. You will notice that this webinar is browser-based. So if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask our speaker questions. You can do that via the questions widget on the left-hand side of your screen. Just type your question in this box and click Submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. I would also like to invite you to fill out a short survey, which you can find in the middle of the screen. If you require any assistance, please use the yellow Help widget. In addition, you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. And now, please allow me to welcome Susan. Over to you.
Thank you, Eugenia, for that kind introduction. Today, you will learn how to discover and optimize the solid form of your API. To begin with, you'll hear a little bit about the company Solvias. Then you will learn why the solid form of an API is important. And then we will discuss how to screen for salts, co-crystals, and polymorphs, how to really understand and select the best solid form, how to develop a crystallization process to produce that form. And finally, we will end with 2 case studies. So Solvias is a global contract research organization that was founded in Switzerland in 1999. And we have over 800 employees now. We are ISO and GMP certified and have been successfully inspected by the FDA multiple times. Our headquarters are in Kaiserauch, Switzerland, and we also have nearby sites in Basel, Switzerland, and Hamburg, France, as well as more recently sites in the Netherlands and in the United States. And this slide shows an overview of some of the many different types of things that Solvias does. Today, this webinar will focus on the polymorphism, salts, and crystallization work, and the interface of that with our custom synthesis and catalysis. Our solutions span the entire life cycle of the product, and the solid state work I will talk about today is focused on the preclinical and clinical phases. But we also have a team that does GMP release analytics, so also making sure you have the best, the correct solid form as your substance and in the product. How do we go about choosing the solid form? When you think of an active pharmaceutical ingredient, a drug, you often think of the molecule, so maybe of aspirin or ibuprofen. But actually, for oral solid dosage forms, what is also very important is the crystal forms that these molecules can self-assemble into. And polymorphs are simply different crystalline forms of the same API. So you can see these blue bricks represent your API, and they crystallize in different ways. And I will talk about why that's important in the next slide. Another form of that an API can have is the amorphous form. Unlike a crystalline form, an amorphous form has no long-range order. And amorphous forms can be developed, but they are inherently thermodynamically unstable, so special formulation efforts need to be made to make sure that they don't crystallize. That's outside the scope of today's webinar. Other crystalline forms can be multi-component systems. So What you can have is you can have a host-guest complex between your drug molecule and a solvent, and that can be either an organic solvent that is used in crystallization, and that's a solvate, or it can be water, in which case it's known as a hydrate. And you can have different solid form, basically different polymorphs of solvates and hydrates. You can have also stoichiometric solvates and hydrates, where there is a defined ratio between the solvent molecules and the API. Or you can have, in particular in the case of hydrates, channel hydrates, so structures with channels where the molecules can diffuse in and out. And these can be complicated to develop, but you'll see an example later on. So you can have polymorphs of solvates and hydrates. Another option is a co-crystal. And again, this is a two-component system, but the difference from a solvate is that the other component, the non-drug component, is a solid at room temperature. Now, you might think that this is just a semantic difference, but it's actually more than that, because if you have a solvate with an organic molecule, it's not going to be thermodynamically stable. At some point, the organic molecule is going to evaporate, but a co-crystal can be thermodynamically stable. And then you have salts. Salts are another two-component system that you might be familiar with. In this case, you have basically an acid-base reaction, so a proton transfer from one molecule to another. This is, again, a two-component system, but there's a bit of a change then in the API because it's protonated or deprotonated. And you can have polymorphs and solvates of them. You can even have combinations. You can have, for instance, a co-crystal of salts that's also a hydrate. So you see there are many possible solid forms that your API can appear in. And you might be asking yourself, why does this matter? Why does anyone care about this? And the reason is that these different crystal lattices correspond to different free energies, and that translates into different physicochemical properties of these different solid forms. So one of the most important ones, you could have very different solubility between the forms, or different dissolution rates. And this is correlated directly with the bioavailability, so how well the drug is taken up by the body. You could have differences in melting points or differences in the chemical stability. And this can play a role for shelf life or supply chain logistics. And particularly in this globalized world where maybe your intermediate or your API is produced in one country and it's made into formulated as a drug product halfway around the world, you want to make sure that your material is stable. Characteristics such as density or hygroscopicity or compressibility can be important, for instance, in manufacturing and making tablets or other solid dosage forms. And hygroscopicity in particular, which also influences shelf life, can influence how you, a lot of downstream processes. So if you have a hygroscopic drug, meaning it tends to suck up water from the atmosphere, then you might have to protect it in a special blister package, or you might have, you could be concerned also just about how to manufacture it, or you don't want to use downstream processes such as wet granulation. And there are other properties as well, and all of these taken together offer an opportunity for intellectual property protection. So if you can find a solid form with better properties than the initial form, you have a chance to get a patent. And this webinar will focus on polymorphism in the pharmaceutical industry, but it's also important in other industries such as pigments, dyes, and even explosives. This pyramid shows our approach here at Solvias to finding the optimal solid form. So we work from the bottom up, from screening to understanding the form, to developing a way to produce the form, and finally producing the form on a larger scale. So let's talk about the different types of screening options we have: salt, co-crystal, and polymorphism screening. And one of the questions you might be asking is, how do you decide which form to develop? Do you want to develop the API itself, or do you look for a salt or co-crystal? And a good starting point is to just characterize the properties of the first solid form you have. And if they're favorable, particularly properties such as solubility, then you might want to go directly into a polymorphism screening of that API. If they're not favorable, the next step is to see, could you make a salt? So does your molecule have an acidic or basic functional group that could potentially react with the salt forming agent? And if so, then you'll want to do a salt screening. If not, you still have the option of a co-crystal screening. And I should add, you sometimes might want to do a co-crystal screening even if your API or salt has favorable properties, because this could increase the range of your intellectual property protection. You also have the option to, there are also regulatory benefits. So the FDA treats a co-crystal as basically similar to another polymorph from a regulatory perspective, so that can have advantages as well. And once you found the form that you want of salt or the co-crystal, then you would need to do a polymorphism screen on that as well. And of course, these screenings can be done in parallel, depending, particularly if your timelines are tight. So I said the first step was physicochemical characterization. What are some of the methods we would use for that? And the gold standard really is X-ray powder diffraction. And the reason is because X-ray powder diffraction directly measures properties of the crystal lattice. So different crystal lattices will have different powder diffraction diffractograms. And this is really essentially the definition of polymorphism is different crystal forms. So this is a very direct measure, but there are other methods that are interesting as well. One of them is Raman spectroscopy. This is a vibrational spectroscopy that is complementary to FTIR spectroscopy. And being a vibrational spectroscopy, it gives you information about local order rather than long-range order. So it's not as direct a measure of polymorphism as X-ray powder diffraction. But because when your molecules pack together differently in different lattices, that will affect their nearest neighbor interactions. So although the differences are subtle, you can generally recognize polymorphs from Raman spectroscopy. And what's particularly, where Raman is particularly useful is it requires no sample preparation. And also glass, quartz, and water are very poor Raman scatterers. And this gives you the possibility to measure Raman spectra through a glass or quartz container without any background interference, which is very useful. Another important method is thermogravimetry coupled to FTIR spectroscopy. This is a simple method. So you put your sample on a microbalance and heat it up and watch the mass loss, but you couple the volatiles that are released to an FTIR spectrometer so that you can identify them. And this allows you to tell, do you have a solvate, do you have a hydrate, or is your sample degrading? Then we have differential scanning calorimetry and dynamic vapor sorption. I will talk about those in a bit more detail later on in this webinar. And finally, there's a range of other methods, standard methods just to confirm purity and chemical integrity, and also to look at the morphology of the crystals that you have on different lengths of scales. So our approach to polymorphism screening, and what I'm going to talk about now is sort of what are the important things you can do. We don't do this every time, and I'll talk about that as well. So after you've characterized your starting material, your goal then is to use a wide variety of solvents and a wide variety of crystallization methods to explore the largest possible parameter space and try to find all the important solid forms that exist. And you do this using sort of classical crystallization techniques like cooling crystallization, which you may remember from an organic chemistry class, or evaporation. You also can use precipitation with an anti-solvent, and you can either add the anti-solvent to your solution or add the solution to a bath of anti-solvent. You can do vapor diffusion, which is basically a very slow precipitation experiment that goes through the gas phase. You can use suspension equilibration, and this is an important technique. So this involves just stirring a suspension of solid in a solvent system. And at a given pressure and temperature, except at exact equilibrium points, basically only one solid form is going to be stable in a solvent system at a given pressure and temperature. And so this allows you to understand relative stabilities of different solid forms. Desolvation is another important approach. So if you have a solvate or a hydrate, you want to try to dry it under vacuum or with heat or with both and remove the solvent. And of course, sometimes the sample could just collapse, but often this can lead to new solid forms. Exploring crystallization from the melt, so without solvents, are interesting, as is sublimation. And if we start some of our experiments from the amorphous form, we can access metastable forms. Again, some of these forms might not be forms we want to develop, but it's important to understand how, that they exist. And in a typical screening, we would also vary the composition of solvent systems to form hydrates. So we would vary the water activity or the water content in different experiments to see, do we have monohydrates, dihydrates, what are their stability ranges, et cetera. And temperature is an important parameter, not only crystallization from the melt, but also suspensions at different temperatures. And we also look at mechanical stress, and this is done In co-crystal screening, actually liquid-assisted grinding is a way to make co-crystals, which we'll talk about co-crystals more in a few minutes, but also you can get an idea if your sample will be sensitive to potential tableting procedures or mechanization. And then once you have all these new solid forms, you have to characterize them. So you need to understand what they are they thermally stable, are they hygroscopic, etc. And what's also important is figuring out which form is stable under which conditions. So if you have a pair of polymorphs that can be monotropically related, meaning one form is always more stable than the other at all temperatures, or they can be aniotropically related, which means you have a crossover point, and below a certain temperature, one form is stable, and above, another form is stable. And this can be relevant, of course, if your transition temperature falls somewhere between the ICH guidelines of 25 and 40 degrees, which we've seen, then you won't have thermodynamically the stable form in both cases. You might have some kinetic stability. And this can also, knowing these kind of transitions can help you decide when to seed in a crystallization process. And what I've shown you here is sort of the general approach, but of course, our projects are fit for purpose. It doesn't make sense to do all of this on something just coming out as a medchem lead. So if you have a very new molecule, you would maybe start just with some risk analysis. Is polymorphism going to be an issue? As it progresses a bit further, it's important to identify the thermodynamically stable form at room temperature and important solvates. And later on, as you advance into the later phases of your project, you want to really have a full understanding, also because in a purer batch, you might have gotten rid of some impurities that hindered formation of certain forms. And then you can also look at your intellectual property profile. And on the right, I've just shown a type of schematic that is something that we might get out of our projects, where you see different salt anhydrous forms, how they relate to each other, different hydration states, how you go back and forwards, maybe even multiple solvates with the same solvent. So that's a polymorphism screening, and that can apply either to the API, a salt, or a co-crystal. But you may wonder, why would you want to make a salt? And the most common reason is improved solubility and therefore improved bioavailability. And if you look at this diagram on the right, on the far right at the high pH values, that's the solubility of the free base, and it's very low. But then you see that there are 4 different salts that are made. They're labeled A, B, C, and D. And you can have orders of magnitude improvement in solubility. So making a salt really allows you to have a large change in parameters. Often you can get better stability. So this could be thermal stability. This could be chemical stability. Perhaps the salt protects a site. And salts are also a useful purification step. So if you have an amorphous API, you can make a salt maybe to purify it. Forming salts of intermediates can be interesting as well. Sometimes you can also improve properties, such as decreasing the hygroscopicity. This is not always the case, of course. Or maybe in some cases, the salts might be more reproducible and a less complex polymorphic landscape than the API. Another interesting possibility is taste masking. So if you have something that's going to be dosed as perhaps a suspension and it's a rather bitter or sour taste, you could think about making a salt with something more pleasant, such as citric acid or saccharin or vanillic acid. And finally, intellectual property protection. If you can improve the properties of your API by making a salt, you can patent it. But it is an extra step, and that means extra work in the synthesis, extra analytics in the end to make sure that you have the correct salt, and it can sometimes lead to inferior properties. If you have a large salt former, you will get bulkier tablets or other forms, and also some characteristics won't be improved. So hygroscopicity, for instance, hydrochloric acid salts are often quite hygroscopic. So there's no guarantee of improvement. And why would you make a co-crystal? Well, there's a simple reason that not all APIs have acidic or basic sites, so a salt is not always an option. Or maybe the APIs or salts have undesirable properties, or you just want to expand your intellectual property or find a sort of regulatory shortcut rather than making a salt. But again, it's an additional step, and there's no guarantee ahead of time that the properties will be improved. You really have to look at this on a case-by-case basis. And the way co-crystals are held together are through non-reactive intermolecular interactions known as synthons. And these are often hydrogen bonds. And these hydrogen bonds you can either have a heterosynthon, meaning you have 2 different types of functional groups connecting, or a homosynthon, meaning you have the same type of functional group connecting between your API molecule and your co-crystal former. You can also have other interactions such as pi stacking that help your co-crystals pack into a different structure. And there are at least 11 pharmaceutical co-crystals on the market. And if you look at this table, you can see a lot of times they have improved characteristics, such as better thermostability, decreased hygroscopicity, or improved solubility, and therefore improved bioavailability. And you can see sometimes, a lot of times we have sort of typical co-crystal formers, can be amino acids like proline, or they can be salts from the FDA's generally recognized as safe list, such as citric acid or fumaric acid. But what's interesting, if you look at Entresto, and about the middle of the table here, it's a co-crystal actually of 2 APIs, or more specifically, 2 salts of APIs, and it's also a hydrate. So this has a lot of complexity in it, and it had improved bioavailability. There was the chance to dose the 2 forms together, and it's used as a treatment for heart failure, and it actually has reached blockbuster status. So co-crystals are part of the reality now. So how can we search for solid forms? And one tool that we have is high-throughput screening. And what I showed you here, this is one of the systems we have at Solvias. It's a robotic system that's been customized for our purposes. And we can make 96 well plates, and then we can analyze them either by X-ray powder diffraction, so we get a diffractogram from every single well on the plate, or we can analyze them with Raman microscopy, where we search out crystals, can image them, and get the corresponding Raman spectrum. And we can even do both on the same plate. And with this robotic system, we can also do a range of experiments in parallel. So we can start with a mother plate of slurries, we can heat them up, and then use the hot mother liquor as a starting material in 3 daughter plates for cooling crystallization, evaporative crystallization, and precipitation. I say that high-throughput screening is a tool for solid form screening because it's important to realize that even though if you run these 4 plates, you have 384 experiments, you don't get all the information about the properties. What you find are interesting parts of the parameter space where you see new solid forms, but then you need to go to the lab, make more of the material, and characterize it in detail. And this here is a photo of an example of one of the X-ray plates. It's covered with a Kapton foil to protect the samples and prevent cross-contamination. And so if you were to do a screening on this, you would record a powder X-ray diffractogram for each well. Then you would basically eliminate the ones that are fully amorphous, because with X-ray, you can't really tell too much about that. Some of these examples here are partly amorphous, but they have at least a few crystalline peaks. And then you cluster them into groups. So here you can see, for instance, the top row, they all have the same peak positions. There will be variation in intensity due to orientation effects, but you group them into clusters, and then you compare them with the references. And as you'll see, the top row, those are all polymorphs of the free base itself, the bottom row. But in between, you see some leads. You see some new solid forms that don't correspond to either of the reagents. And this is what I showed you here as a salt screening, but this is particularly useful for polymorphism screenings, because a polymorph is by definition, it's a system with, you have an API, you don't have a coformer, and it's crystalline. So X-ray can sort out the amorphous things quite quickly. We have the other option, which is analysis by Raman microscopy. And here is a plate for an example like that. And what you would get here then are different images from the different wells. And you can actually correlate the crystals or the particles you see with the spectra. And again, you would do a classification like I showed in the past. You would cluster the spectrum and compare it to the references. But what can be useful here in Raman is particularly in salt and co-crystal screenings. Sometimes you will get an amorphous sample, such as the second picture from the top. And with X-ray, you just see that it's amorphous. But with the Raman spectrum, you can sometimes see subtle shifts that indicate that the components have complex. So they've made an amorphous material, but there's a hint that they interact. And that gives you a possibility to optimize and crystallize later on. So after we found these forms, the next step is to characterize them and understand them. And we go through various criteria. So if you're comparing a salt to the free form, you definitely want to look at the solubility, see if you have an improvement, since that's often a reason for making a salt or co-crystal. You'll also want to make sure that this new form is stable, and possibly in some preformulation work, test its compatibility with different excipients. You want to make sure that it's not too hygroscopic, and you want to look at the habit or morphology, because long needles or flat plates can sometimes be difficult to filter, but if you have an equant morphology, that can be useful. Then once you've chosen your form, whether it's the free drug, a salt, or a co-crystal, you need to look at the polymorphs in detail. And one aspect is to look at the stability as a function of temperature. And again, as I mentioned a few minutes earlier, you can have enantiotropically related polymorphs. They're shown in this diagram here, where at low temperatures, form 2, the blue line is more stable, and at higher temperatures, form 1 is more stable. And depending on where this transition temperature is, will influence things like storage conditions or crystallization processes. You also want to look at the kinetics of transformation. So you might have a metastable form that persists a long time, or they might be very labile and not as relevant. Then you want to identify any hydrates in your system. And here you want to look at the critical water activity. And in the diagram in the lower right, you can see this was a publication from our group several years ago. And it's a plot of the stable forms as a function of water activity and temperature. So if you look across a horizontal line, you can see, for instance, at room temperature, the orange part of the diagram that's saying this hemihydrate form E was the most stable at low water activities. And then as you increase the water activity, which also corresponds to increasing the relative humidity, form C, the dihydrate, becomes more stable. And finally, in this case, form A was a pentahydrate, was stable at room temperature from water activities of 0.4 to 1. So from 40% to 100% relative humidity, this was a stable form. And this had a large range and was chosen as the form for development. You also want to see if your hydrates are stoichiometric hydrates or channel hydrates. Channel hydrates are a bit trickier to develop specifications for. And then you want to look at conversion rates and resulting forms. A lot of times when you dehydrate a hydrate, it goes into one amorphous form, and when you rehydrate it, it might not go back into the same form. So you need to sort of understand, and this can be important for drying processes, are the transformations reversible or not? And then, of course, look at the solubility, because ultimately a hydrate's going to be the stable form in aqueous solution, and this means this could put a limit sort of on your bioavailability. And finally, you also need to understand solvates, even if they are very rarely developed, because you want to know which solvents to avoid in your final crystallization process. This also could give you some clues as to interactions that you might see in the formulation. And finally, desolvating solvates can be a way to reach new forms. And there are several examples in the literature, for instance, where the most stable form was accessed by desolvation, first accessed by desolvation of a solvate. So once you, to make some of these evaluations, you use various analytical tools. And one of these is differential scanning calorimetry. This probably many of you are familiar with. Here you can see an example of a starting material that has 2 endothermic transitions. And an endotherm could correspond to a melt. It could correspond to a dehydration, desolvation event, but it could also correspond to a solid-solid transformation. And based on the position and the magnitude, here, that was the hypothesis. Here we had that the starting material transformed into a different form, and the melting point actually corresponded to the new form. And we were able to confirm this by doing variable temperature X-ray diffraction. So we have an X-ray diffractometer, and we can heat the sample up to different temperatures. So if you look at the bottom line in blue, at room temperature, you have one form. I've just shown part of the diffractogram. As you heat it up to 120 degrees, you see slight shifts due to thermal effects on the crystal lattice, but it's the same polymorph. But then if you go up to 225, which is on the other side of this endothermic transformation, you see a different solid form. So this confirms that you have enantiotropically related forms. As you lower the temperature to 120, you see a mixture of the forms. And finally, going back down to room temperature, you're essentially back at the initial form. So in this case, the high temperature form was not very kinetically stable, but nonetheless, we were able to confirm that we understood what was happening in the DSC. Dynamic vapor sorption is another very useful method. And this is also a method where you take a microbalance, put your sample on it, and expose it to different relative humidities, either in a ramped or in a stepped manner. And you can understand things about different hydration states. So this diagram here is the real customer sample, and they started out with the dihydrate. And as we lowered the humidity, it converted into a sesquiohydrate. And as we lowered it further, it converted into a monohydrate. As we lowered it further, we got an anhydrous form. Then as we raised the humidity, we see that it bypassed the monohydrate and converted directly back into the sesquiohydrate, then at higher humidities into the dihydrate, then at even higher humidities, we found a trihydrate of this form. And finally, at the end, going back to 50% relative humidity, we're back at the dihydrate. And you'll see there's a hysteresis in these transformations, and this is a kinetic effect because basically, except at a transition point, thermodynamically only one of the forms would be stable. Now, as you can imagine, such a complex diagram, it would have been a nightmare to try to develop just one of these forms. And this led the customer to do a salt screening, and we successfully found a new salt that was not so complex. Here on the right-hand side, you might see a more typical hydration-dehydration process. Here, what you have is a monohydrate form. And if you look at the blue curve, that's the humidity ramping. So raising and lowering the humidity, you see that basically the mass change is very stable over a large range. And only when you get down to below 5% relative humidity does it convert into the anhydrous form. And then there's a certain degree of kinetic stability. The anhydrous form persists a bit and then converts into the monohydrate. But then you're going to want to know, okay, somewhere in this range is thermodynamic equilibrium. And you can find this by doing competitive suspension equilibration experiments. So you mix the two forms together at different water activities and equilibrate them until only one form persists. And then you can see what the stability range is. And such experiments showed here that the critical water activity was somewhere between 0.17 and 0.21, so corresponding to 17 to 21% relative humidity. So below 17% relative humidity, the anhydrous form was stable, and above, the monohydrate was stable. And so then it was very clear in this case that the monohydrate would be the form developed. And we can also use X-ray diffractogram, this time in situ varying of the humidity, to understand dynamic vapor absorption. And here you see again a case with several steps. And on the right-hand side, you see the variable humidity powder X-ray diffractograms. So we started at about 45% relative humidity. Then in the X-ray instrument, we lowered it to 2, and that corresponds to here at the very lower left of the DVS, we found an anhydrous form that had not been previously discovered. Then if we raise the humidity to 11%, corresponding to this jump, this step in the DVS curve, we found a form that was designated as hydrate 1. And this was very interesting because it led us to realize that our original starting material was actually a mixture of forms. It had hydrate 1 as a component, but was also something else. And then raising the humidity to 29%, you saw the same mixture as initially. And by the time we got up to 49%, you saw pure hydrate 2. So this shows you how you can work together not only to understand hydration states, but sometimes see if you have a pure phase or a mixture. So after we've done the type of characterization like that, the next step, we choose our solid form. Now we have to make it. We have to make it on a larger scale. This is where crystallization development comes in. So this is just an example of it's a concentration versus temperature. The blue line is the solubility curve. And to the right side of the blue line, at higher temperatures, the system is undersaturated. So you're not going to have any crystallization. And if you go to the left of the blue line, so lower the temperature for a given concentration, you will have a supersaturated solution. And above a certain point, you get uncontrolled spontaneous nucleation. So this is called the Labal region. And what you have in between these two regions is the so-called metastable region. And here you have some supersaturation, so you can crystallize, but there's not such a strong driving force for spontaneous crystallization. And so you can develop a seeded process where you lower the humidity seed. And if you stay in this metastable zone, you can get a controlled crystallization of your desired form. And we can measure the stable zone with using turbidity probes, or also 2 other probes that you'll see on the next slide. And the concept is quite simple. You start with a high temperature, you have a solution, and you lower the temperature until you observe precipitation. The solution becomes cloudy. It's a so-called cloud point. Then you raise the temperature again. You have this cloudy solution, and you note the point when it dissolves again, the so-called clear point. And the dissolution points are on the solubility curves, and the cloud points represent the edge of the metastable zone. And then you can dilute the system and repeat the process. So lower the temperature again at this lower concentration until you see the cloud point, and then raise the temperature again until you see the new clear point and dilute again. And on the right here is an example of just a customer's real system where we did one of these metastable zone widths, and you can see the solubility and the metastable, and this was about a 25-degree width to the curve, so that gives us quite a bit of room to develop a robust crystallization process. The tools that we would use for such a process are shown here. We have other scales as well, but here are two 400 milliliter jacketed glass reactors, and we have some probes, and one of them is the Easy Viewer from Mettler to Toledo, and that actually looks inside the system and records images in situ. So during your whole crystallization process, you can actually look in the system and see what happens. You can see changes in size and shape or agglomeration or breakage. There's also the focused beam reflectance measurement probe. So this is a system for measuring chord length distributions. What happens is you have a laser that rotates, and as the path of the laser intersects with the path of the particle will reflect the light back, and the length of the pulse that results is basically one dimension of the particle. So the distributions you get are not exactly the same as you would get from a laser diffraction particle size distribution. But what it does give you is a way then to follow your crystallization process. So if you have a distribution and during the process it's moving toward larger sizes, then you see you have growth. If you have a sudden appearance of fines, you have nucleation. And using this kind of feedback, we can vary parameters, we can control the dosage and the temperature and the stirring rate, and develop a controlled crystallization process. And once we have that process, we can transfer it to our kilo lab. So we have an ISO and GMP kilo labs where we can produce under GMP in up to 100-liter vessels. We have some stages in between. And we can really do a smooth transfer of what we learn from the crystallization optimization on the small scale to the larger scale. And this can be directly integrated into an optimization and development of a synthesis and perhaps a catalyzed synthetic process. So that not only do you get the right material with the right purity, but you also get it in the right solid form. And the GMP material on the kilogram scale can be used for clinical trials with humans, or for instance, as seed crystals in a pilot plant. So with the remaining time here, I would like to just go through two case studies. And the first is a co-crystal high-throughput screening. And in this case, we had a neutral API with an undesirable morphology, very long needles, a high aspect ratio, and we screened various solvents, but there was no way to crystallize this directly in any other morphology. And so because it's neutral, there were no acidic or basic sites, we couldn't form a salt. But there was an amide group, and there is the possibility, and that's shown on the right here, of perhaps making a heterosynthon. So we might be able to couple it to some carboxylic acid. And there were other interactions we looked at as well. So we selected 24 potential co-crystal formers and did a high throughput screening. And the first screening was done by mixing roughly and evaporating from 4 solvent systems. And evaporation is very good when you have a symmetric phase diagram. So when the solubilities of the two components are similar in the solvent, then that gives you a good way to access the co-crystals. And we then analyzed the products by Raman microscopy. And then we resuspended these residues as very concentrated slurries. And so when it's concentrated, basically you're at the bottom of the curve for solubility. So it's similar to what you would access with liquid assisted grinding. And then we can find co-crystals both from symmetric and asymmetric phase diagrams. And again, we analyzed the products by Raman microscopy. And these are the results of the evaporation experiments. These are the images. We classified the Raman spectra then. And from evaporation, we found 10 distinct hits, 2 different ones with succinic acid. And in this picture, the solid green lines represent unique hits, and the dotted lines are just cases, other wells in which those same hits were also observed, because we had 4 different conditions for each potential co-crystal former. And then we did similarly for the suspension equilibration, and we found 8 distinct hits in that plate. And comparing the 2 plates together, because some were found in both, we had 13 unique leads. And then together with the customer, we decided to scale up and characterize 6 of these leads. And the results can be seen in this table. What you have are different characterization methods, the 6 co-crystal formers, the best example of each, and on the far right, the properties of the best form of the free drug. And then you can go through and start looking at things. So in 3 of the cases, we were not able to obtain a fully crystalline sample. It was partially amorphous. Maybe it would be possible with more work, but it's at least a little more challenging. So those are suboptimal. Looking at the TGFTIR, 1 sample was a solvate, another decomposed at low temperatures. Again, maybe there's another polymorph with better properties, but we have better options. Then looking at elemental analysis, we saw that while we had a nice stoichiometric ratios with benzoic and maleic acid and ethyl maltol, 2 of the samples did not comply. And at this point, we decided not to consider the 3 formers on the right, but just focus on the first 3. And we can see, for instance, maleic acid had a melting point that was 10 degrees higher than that of the free drug. And looking at the dynamic vapor sorption, the free drug and the maleic acid co-crystal were slightly hygroscopic, but the benzoic acid one was non-hygroscopic. The ethyl maltol decomposed, so that eliminated that co-crystal former. All 3 of them had suitable solubility. And in the end, both the benzoic acid and the maleic acid had mixed morphologies. So they didn't only have those long needles anymore, but there was a chance to really create a more equant morphology. And so in the end, this screening gave the customer the choice of 2 co-crystals with superior properties to the free drug. The final case study that I will present is related to crystallization process development. And here the customer came to us after having already done a fair amount of work. They knew a bit about the polymorphism, and what they found was form 2, which was a channel hydrate, and form 3, which was an anhydrous form, often crystallized concomitantly. So they often got mixtures of the 2. And they what was also interesting was they noted an exothermic enthalpy of solutions, heat of solutions. So in some solvent systems, you actually had a lower solubility as you raise the temperature rather than a higher, which means instead of doing a cooling crystallization, you would do a heating crystallization. And the method that they had for form 2 involved the surfactant and emulsifier tween 80, and it was a heating crystallization. And they also had a method for form 3, involved a mixture of ethanol, water, and a liquid polymer, polyethylene glycol, with a molecular weight of 300. It was also a heating crystallization. But the yields of both methods were suboptimal. So they wanted improved processes, actually for both of them, but I'm going to talk in the rest of this webinar just about the case for form 2, because that was the form that was ultimately selected. And they wanted crystallization processes that were robust and scalable and transferable. They wanted a volume efficiency of less than 40 volumes. That might not seem so stringent. A lot of times 10 is a more common goal, but this is sort of, they wanted better than what they currently had. They also wanted filtration and drying times. Again, these seem like very unusually long times, but because they were using these solvents that were just had very low vapor pressure, so they were using this surfactant and polyethylene glycol, basically wanted a significant improvement over their current process. They wanted polymorphic control and chemical purity and small particle sizes. As I said, they ultimately selected form 2, so that's what I will focus the rest of this example on. And what we first did was we looked at some low molecular weight solvents, some standard ones, but we did not get form 2 or form 3 from those. You formed anything where the solubility was sufficiently high, you formed a solvate, and there were a lot of times you would observe gels. And so we used a diluted version of the surfactant and also slightly varied the other mixture. And without seeding, we got very inconsistent results. With seeding, we did improve the yield, but the method was still not as reliable or robust as we would have liked. At this point, we decided to adapt the process for form 3 and apply it to form 2 with seeding in some small scale experiments. So these are milliliter scale experiments, and varied solvent ratios and heating profiles and anti-solvent addition rates, stirring rates, et cetera. And then we used the focus beam reflectance measurement on a 20-gram scale and found that it was very important to add this API portion-wise with very vigorous stirring. And we were able to come up with a process that we could transfer to the Kilo lab on a 2.5 kilogram scale in a 100-liter reactor. And it worked very well in the sense the crystallization went smoothly. There was no problem to filter it. We got the correct polymorph, and it was reproducible. It was run 4 times on this kilo scale without a problem. The only problem was that there was a residual PEG 400 content of 2.5 weight percent, which was too much, but we had to be careful. We couldn't dry this sample too much because, if you remember, it's a channel hydrate, so we didn't want to dehydrate it and get into another form. So this took us back to the lab where we optimized the washing procedure, looked at different solvent mixtures, temperatures, protocols, et cetera. came up with a procedure that was successful on the lab scale, but was not as successful on the 2.5 kilogram scale. So there was still 1.6 weight percent of PEG 400, a bit better than 2.5, but not good enough. And at this point, we took a step back and said, okay, are we really stuck with using PEG? Is there not another solvent that we could use? And so we did various measurements of exact solubilities and solvents and mixtures at different temperatures. But we were able to confirm acetonitrile led to a phase separation. We got solvates with dioxane and ethanol, and a lot of the other solvent systems, there wasn't solubility. But what we noticed is that the ethanol solvate would spontaneously desolvate. And when it desolvated, it went to form 2. So the question is, could we maybe use ethanol and water and get to form 2? And the next step then was to determine the critical water activity. So what we did here in this table on the right is we looked at different ethanol water mixtures with different water activities, and we equilibrated the ethanol solvate in form 2 for 5 days to see what form it converted to. And again, thermodynamically, if you're not exactly at the critical water activity, there will be one form. And what we saw was the ethanol solvate was stable up to very high water activities, and there was only a very small range where form 2 was thermodynamically stable. But interestingly, form 2 was still kinetically stable. So in this top solvent mixture, 4 to 6 ethanol water, where the ethanol solvate is stable, if you don't have any seeds of the ethanol solvate, form 2 will remain form 2 for 2 weeks. And so then there's the question, okay, there's enough room there to possibly develop a crystallization process. And there was no strong temperature dependence, so we had to focus on anti-solvent dosage. We then did a series of slurry experiments with various pull points, but found it was not possible to either take the amorphous form or form 1 as the starting material and transform them directly into form 2. But what we could think about doing was using the ethanol solvate as an intermediate. So the idea is you take one of your forms as the starting material, you put it in an ethanol-rich solvent mixture, deliberately form the ethanol solvate, and then add water as an anti-solvent and try to either convert it into form 2 in solution or possibly through the washing and drying process. And what was challenging was that we found there was some phase separation and then precipitation of a gel-like substance. And we tried this at different temperatures, but it didn't help. But nonetheless, these first trials showed that we could reproduce it on a 20-gram scale with acceptable levels of residual ethanol. But because the process was slow, because we had these gels and other intermediate phases, we needed to optimize it more. And what you can see here, this is an example of an FBRM readout. So the orange line is showing you the particle count. And after you seed, the particle count goes up. And at the peak, you get a milky suspension. But then at some point, as you continue to add water, the particle count goes down and you start to see a gel-like, a sticky precipitate forming on the walls of the reactor. And this is also the case as the particle count goes very low. And then at some point with enough water, you got good behavior and you had a very nice, nicely stirrable white suspension. So how do we avoid that? And we did a lot of small milliliter scale experiments looking at solubility versus temperature. And then we did the FBRM experiments. And what we found is we had to reduce the concentration to eliminate phase separation. You don't generally want to reduce the concentration because this lowers your volume efficiency, but sometimes it's necessary to get a controlled process. And we slowed the process down. We tested temperature cycling. And what we saw here, we saw this very interesting FBRM signature where after you seed, the particle count drops, but we did actually see white material forming. So we had an unusual case of something where the FBRM probe was not able to pick up the particles so much, but we could show that there was a white suspension with no gel. And so then we worked on optimizing the process. One good way to do an anti-solvent dosage rather than to just dose at a consistent rate is to use a parabolic profile. The idea being that you add anti-solvent very, very slowly when your system is highly saturated, and then as more and more precipitates out, you can add it at a faster rate. We approximated this parabola with 4 linear ramps. But what we found is it was useful to slow it down. So in between each of these ramps, we had a holding time, and that gave the system enough time to really prevent any supersaturation buildup and made the process more controlled. And we were able to identify the critical stage. And if you look here on this curve, you can see the green steps are the anti-solvent dosage. We took in-process controls, and we could see the first 3 were ethanol solvate, but then we had a smooth transition to form 2 where we actually see that the mean particle size increased. And so avoiding a gel, we were able to produce form 2 in solution. And then there was additional optimization with the seeding point. We played around with temperature cycling, but it didn't help. In the end, we got this final process listed here, where we had these holding times. And what we got was form 2 with basically a quantitative yield, high crystallinity, small particle size, the amount of water we would expect for a channel hydrate, and no residual solvent, organic solvent. We could then scale this up to the 100-gram scale in a 5-liter vessel to make seeds, and scaled it up to the 2.1 kilogram scale. Again, got excellent purity and yield. Particle size was a bit larger than we'd like, but that could be mitigated by micronization. And the process was basically the same as developed in the lab. We had to adjust the stirring rate so that the energy dissipation was matched, and there were some slight differences in the drying, but the process was good. We could filter it quite quickly, 105 liters in 30 minutes, and we achieved a successful and reproducible kilogram scale crystallization process. So to conclude, when should the solid state be studied? Basically at every point of development, but for different reasons. So early on, trying to decide which solid form to choose to develop. Then as you get further along, as your API progresses through the clinical stages successfully, you want to look at the polymorphism again to make sure you didn't miss anything, that you've covered the intellectual property. And then you want to develop the crystallization process that you will use, but then throughout it be used for troubleshooting later on when the product goes on the market, which shouldn't happen if you've developed your process correctly, but sometimes customers come to us at later points in time. Release analytics to make sure you want the form you would like, or further intellectual property. So if you choose the right solid form, I hope you've learned today that you will get a better product, you will get intellectual property protection, you will get a process that is faster, more robust and reliable, efficient and cost effective. And what we at Solvias can bring you is a lot of experience and support for all phases of development, and also flexible solutions that are tailored to your needs. And through the interplay of our different teams and our extensive analytics, which I did not go into in much detail today, we can help you develop your solid form. And if you'd like to learn more, I can recommend this book. This was edited by Rolf, who is now partly retired, but worked at Solvias, and 3 of the chapters here are written by members of the Solvias team. So I would like to now thank you for your attention and take any questions you might have.
And thank you very much for this presentation. Just a reminder for the audience, to ask questions, you can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar, and they will be answered at a later date. And it looks like we've got several questions already. And our first question is, are you able to determine the amorphous content in crystalline samples?
Yes, that's a good question, and that's something that's really done by our GMP specialized analytical team. But when we have crystalline samples, I mean, you could do this by DSC, which I discussed today, but there are actually 2 methods that are quite sensitive, isothermal microcalorimetry and solution calorimetry. And our thermal lab has a lot of expertise in really developing the calibration curves and getting down to limits of detection of amorphous content in a drug substance of under 1%.
Okay, thank you very much. And our next question is, do you use in silico methods for co-crystal prediction?
Okay, that's a very good question. It's actually one that I anticipated. So I have an extra slide here. Of course, everyone is talking a lot about in silico and can you predict co-crystals. And we had a PhD student working together with us and with some universities exploring co-crystal prediction using CosmoQuick and molecular complementarity. And the results were quite good. I mean, he was able to discover and scale up new co-crystals. But it's important to realize that the match isn't perfect. So actually, the number 2 ranked co-crystal could not be produced despite extensive efforts. Some of the intermediate and some of the ones that were quite low ranked actually could be produced. So while there is a place for sort of in silico methods as a guide, I think we're quite competitive with empirical screening. And in the end, you still have to figure out how to make the substance. And so there will always be a place for empirical work in the lab to make the form, but it will be exciting to see in the next few years what comes out of sort of further in silico techniques and how they will work together with experimental work.
Okay, thank you very much. And how many patents does Solvias have on solid forms?
We have approximately 100 patents that have been granted. There have been some other applications that are submitted over the years. And I should add that, of course, on a patent, the discoverer of the form needs to be mentioned on the patent, but all the intellectual property belongs to the customer. So the patent belongs to the customer. We get no financial remuneration, but we do, of course, contribute to finding the new form, and we also can consult and support. We have a lot of experience in putting together sort of the right type of results and how to present them to work with patent lawyers to help you get your API patented.
Thank you very much. And our next question is, how are projects managed at Solvias, and why should we work with you?
So at Solvias, our projects, we have a project leader who's a single point of contact who manages the project, but you have access also to the experts. So if at some point we need to develop an analytical method, then we will get the expert on the telephone call with you. We have weekly or bi-weekly telephone conferences, but that you can really take advantage of their information. And our project leaders serve also as consultants, and I think it's also important to realize that we try to be flexible. So we have modular quotations, but in the end, we don't try to fit your compound into our scheme. We try to figure out how we can serve you, how we can find the information that you need. And so we have this very interactive flexibility. It's really like a partnership. Not just a service.
Perfect. Thank you very much. And we have time for one more question. Do you perform HTS PXRD on the same plate? Does the material of the plate have an effect, or do you perform PXRD in transmission geometry?
We're currently performing the sample that I showed you there is in reflection geometry, but we have, this is basically we have a metal base that is basically does not have a strong signal in either X-ray or Raman, and the Kapton foil in the X-ray is also something that does not have an X-ray signal. So we can do it on the same plate. We also have options of different plates.
Okay, thank you very much. And just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. I would like to thank Susan for this presentation and Solvias for sponsoring this session. To the attendees, you will receive an email telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is pmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.